and welcome to my gut microbiome presentation. A microbiome is the combined genetic material of the microorganisms present in a particular environment. A gut microbiome is the complex community of microorganisms that live in the digestive tracts of humans and animals. In humans, bacteria detected in the gut microbiome, like butyrate-producing Faecalibacterium and Copernicus, have been indicators of living a higher quality of life. This means that the possibility of having depression or being depressed is less likely than those who do not have these organisms present. Bacteroidetes and firmicutes are two of the four phyla that is very apparent in the human gut microbiome. Faecalibacterium, or F. presunti, which is found among firmicutes, is very abundant in humans, making up about 8% of the gut microbiome found in the colon. This bacteria plays an important role in keeping our gut healthy. Researchers have found that those who suffer from Crohn's disease, IBS, active ulcerative colitis, and type 2 diabetes have decreased amounts of these microbiomes present. For our experiment, we hypothesized the effect of age on the gut microbiome, and we hypothesized that there would be a significant difference between our two samples. So we chose to compare two dog samples for our experiment. Our first dog was sample 1092, which is a 10-year-old boxer, and our second dog, which was sample 469, was a 2-year-old husky. In week 1, we isolated and purified the DNA using the QIAGEN kit, and then we concentrated the DNA using the Zymo kit. In week 2, we amplified the 16S rRNA gene, and we quantified the DNA of the two samples by using UV absorbance spectrophotometer. We recorded the concentration as well as the 260-280 value. The 260-280 value determines the purity of the DNA. In week 3, we checked the products of the PCR reaction by using the Argos gel to remove unused primers, DNTPs, and other PCR reagents. The purpose of using the gel electrophoresis was to determine the amount and concentration of our DNA. In week 4, we cleaned up the index PCR reaction using the Agincourt Ampere XP kit in order to remove the primers. We also performed indexing PCR, which tags our DNA. We then repeated PCR cleanup. Later, we quantified the amplified DNA using the fluorimeter and prepared the dilution of the purified DNA in order for it to be sequenced. Our initial UV spectrophotometry DNA qualification results were 6.4 for the nanograms per microliter for sample 1092, and our 260-280 value for that sample was 3.48, and our concentration for sample 469 was 3.3 nanograms per microliter, and our 260-280 value was 1.1. In our gel electrophoresis results, we saw no DNA in either of our samples or the negative. We did, however, see samples DNA in our positive control as expected. So as you can see here, we had lanes 1 through 4. Lanes 1, 2, and 3 did not have any bands present, but lane 4 did, which means that there was DNA present in lane 4, but there was nothing present in lanes 1, 2, and 3. The quantification of our cleaned up PCR amplicon for our samples were too low for both. For our samples, we did not have any successful reads, so we had to choose new samples. We chose sample 321 and sample 344. Both samples are dogs older than the other, one older than the other. We used the results from those samples for the rest of our experiment. First, based on the kingdom, we found that both samples of DNA from the older dog and the younger dog had roughly about the same amount of bacteria. So as you can see here, sample 321 is our older dog and sample 344, which is our smaller dog. So the older dog is the blue and the smaller dog is the orange. You can see that they have roughly about the same amount of bacteria present. So along the y-axis on the x-axis we have the organisms that were detected in each of our samples, and along the y-axis we have the percentage of the amount of organisms found. So the bacteria was about 100% for both of our samples for our kingdom. For the order, we found that the younger dog possessed more variety of bacteria than the older dog did. So you have more you have the same amount of organisms present in both samples, but as you can see here, the orange sample, also known as a younger dog, had more of the bacteria present. For example, the Colostridales had about 30% present in our older dog and about 55% present in our younger dog. Lastly, based on the genus, we found that our older dog had more types of specific bacteria than our younger dog did. So our older dog had about 7 different types of organisms present, while our younger dog only had 4. So our older dog had more of a variety in the amount of bacteria present. So our group was unsuccessful at obtaining adequate DNA samples from both the initial fecal matter samples. The UV spectrophotometer readings showed that both samples were void of viable DNA to continue with the experiment. There were no bands present during gel electrophoresis, but our experiment otherwise went as planned with no discernible cross-contamination. We opted to use verified adequate samples 321, 344 to continue with our experiment. The sample from our younger canine 344 had about 1,304 total reads, and our sample from our older canine, which was sample 321, had about 14,862 total reads. These numbers were sufficient for our purposes. As stated above, our reads from our original samples were not sufficient during the gel electrophoresis process. The Amplicon PCR did not provide us with a large enough quantity of DNA to reasonably continue our experiment, as our samples did not reach the 1.5 nanometer minimum amount that would be viable. Presence of a PCR band at about 550 base pairs in our positive control showed adequate amount to carry on with our initial sample. The absence of a band at 550 base pairs in our negative control proved our experiment did not have any cross-contamination, as you can see here. So, Megamonas ferments glucose into acetate, which was one of the bacteria found in one of our samples, and it basically ferments glucose into acetate and prionates, which is conducive for a healthy species. So overall, looking at our data, we can conclude that our hypothesis is supported and that the age of canines did have an effect on the gut microbiome, as sample 321 and 344 had a variety of different bacteria present. For example, sample 321 lacks any bacteroidetes, and sample 344 lacks the presence of Streptococcus, Escherichia, Collinsella, Sharpia, and Megamonas. Thank you.